champs of Examsnet, welcome to the question series of Examsnet. I hope this series of detailed explanation is helping you a lot. So in today's session we are going to discuss NEET 2017 biology paper and we have a total of 90 questions. So let's start the session. The first question is from the chapter biomolecules and the question is which one of the following statements is correct with reference to enzymes. Okay. So let's see. The first one is holoenzyme equals to apoenzyme plus coenzyme. Then the next one is coenzyme equals to apoenzyme plus holoenzyme. And then we have holoenzyme equals to coenzyme plus cofactor. And the last one is apoenzyme equals to holoenzyme plus coenzyme. So the correct answer will be option number A. Okay. Holoenzyme equals to apoenzyme plus coenzyme. So here the apoenzyme is the protein part, and coenzyme is the non-protein part. Okay. And when combined together, they form the holoenzyme. So option A will be the correct answer. Question number 2 is from the chapter Chemical Coordination and Integration. And the question is, a decrease in blood pressure or volume will not cause the release of. Options are arterial natriuretic factor, aldosterone, ADH and renin. So the correct answer will be option A, that is arterial natriuretic factor. Okay. So it is released in response to an increase in blood pressure, not in response to decrease in blood pressure. Okay. So in short, we write it as ANF, that is arterial natriuretic factor. Okay. It is basically a peptide hormone. What kind of hormone? Peptide hormone that is secreted by the arterial wall of the heart. Okay. And it is secreted in response to increase in blood pressure. Response to increase in blood pressure, or we can say that it is released under high blood pressure condition. Okay. Then the reason that it is released under high blood pressure condition is that it acts as a vasodilator. Okay. Acts as vasodilator and thus it helps in decreasing the blood pressure. Okay. In addition to that, it also reduces the water and sodium concentration. Reduces water and sodium concentration in the circulatory system. Okay. All the other options, that is aldosterone, ADH and renin, these are secreted in response to decrease in blood pressure as mentioned in the question. Okay. So aldosterone is a steroid hormone that is secreted by the adrenal cortex. Adrenal cortex. ADH is secreted by the posterior pituitary. Okay. Posterior pituitary. And renin is released by juxtaglomerular cells of kidney. Juxtaglomerular cells of kidney. So all these help in increasing the blood pressure. Therefore, they are released when the blood pressure decreases. Okay. So option A will be the correct answer. Question number 3 is from the chapter Digestion and Absorption. And the question is, which cells of crypts of Libercon secrete antibacterial lysozyme? Options are Paneth cells, zymogen cells, Kupffer cells and Argentophin cells. Okay. So the correct answer will be option A, that is Paneth cells. Right. These are found in crypts of Libercon. And what are crypts of Libercon? The gland, intestinal gland that is present between the villi of small intestine. Okay. So crypts of Libercon, intestinal gland present between villi of the small intestine. Okay. So here the Paneth cells are found and these help in the regulation of composition of the intestinal flora. Okay. Paneth cells regulate the composition of intestinal flora. The next option is zymogen cells. These are found in gastric glands. Gastric glands, and they secrete which enzymes? Pepsinogen and gastric lipase. Okay. Then we have Kupffer cells. Option C is Kupffer cells. These are the phagocytic cells. Phagocytic cells which are present in the sinusoids of the liver. And the last one is Argentefin cells. These are glandular cells which occur in the lining of the digestive tract. Okay. So these are found in digestive tract and they secrete serotonin. We are talking about the Argentefin cells. Okay. Secrete serotonin. And this serotonin helps in peristaltic movement. Peristaltic movement of the muscles in the digestive tract for the movement of the food. Okay. So according to the question, the correct answer is option A, Paneth cells. Question number 4 is from the chapter Biomolecules, and the question is which of the following are not polymeric? Okay. Options are proteins, polysaccharides, lipids and nucleic acids. So we know that the proteins are the polymers of amino acids, right? Amino acids. So this is polymeric in nature. Okay. Then the next one is polysaccharides. These are the polymers of monosaccharides. Monosaccharides. So polysaccharides are also polymeric in nature. Then lipids are not polymeric. Okay. These are not polymeric. And the last one, nucleic acids, is also polymeric because these are monomers of nucleotides. Okay. If we talk about the composition of lipids, then we know that these are composed of glycerol and fatty acids. Okay. Glycerol and fatty acid. So the correct answer will be option number C, lipids. These are not polymeric. Question number 5 is from the chapter Sexual Reproduction in Flowering Plants, and the question is functional megaspore in an angiosperm develops into. Options are endosperm, embryo sac, embryo and ovule. So we know that megasporangium has megaspore mother cells, MMC, megaspore mother cell. Now this megaspore mother cell divides meiotically. Okay, so meiotic division or meiosis takes place, and as a result 4 megaspores are produced. 4 megaspores. Now out of these 4 megaspores, only 1 megaspore is functional, whereas the other 3 megaspores degenerate. Right? Now this one functional megaspore develops into an embryo sac. Okay. So this is what the question is asked about. This functional megaspore develops into an embryo sac. So option B will be the correct answer. Endosperm, how is it formed? It is formed by the fusion of polar nuclei and a male gamete. Okay, male gamete. And it acts as nutritive cell. That means it provides nutrition to the growing embryo. Okay, nutritive cell. Then embryo, we know that it develops from a zygote. Okay, develops from a zygote. And what about ovule? It develops into a seed. After fertilization. But the question was asked about the functional megaspore. So embryo sac will be the correct answer. Option number B. Question number 6 is from the chapter Neural Control and Coordination. And the question is myelin sheath is produced by. So what is myelin sheath? It is also known as medullary sheath. Okay, medullary sheath. It is basically an insulating layer. Insulating layer that covers the action of the neuron. Okay, which part of the neuron? Action of the neuron. Covers the action. And the function of the myelin sheath is that it helps to maintain a constant membrane potential. Okay, maintain a constant membrane potential. It is produced by two types of cells. First one is Schwann cells, which is part of peripheral nervous system, and the second type is oligodendrocytes, which are part of central nervous system. Okay, so these two cells produce the myelin sheath. Therefore, option D will be the correct answer. Now, question number 7 is again from the chapter Sexual Reproduction in Flowering Plants, and the question is attractants and rewards are required for. Options are entomophily, hydrophily, cleistogamy, and anemophily. So the first option is entomophily, that means insect pollination. Insect pollination, that means the pollen grains are transferred by the insects. Okay. Second one is hydrophily. So here the pollens are transferred by the flow of water. And we know that water is an abiotic agent. Third one is cleistogamy. So cleistogamy represents self-fertilization. Self-fertilization. So here the male and female parts are present in the same flower, and that too a closed flower. Okay. So there is no chance of cross-pollination here. Okay. Also, no external agent is required in this case. No external agent for pollination. And the last one is anemophily, that means wind pollination. So here the pollens are transferred by the wind. Now in the third one, that is cleistogamy, no external agent is required. That means there is no need of attractants or rewards, okay, because these are used to attract the agents. Now the second one and the fourth one, hydrophily and anemophily, in these cases the abiotic agents are involved, okay, so we don't need to attract them, okay. And the first one is entomophily. Yes, here the attractants and rewards are required to attract the insects towards the flowers, okay. So option A will be the correct answer. Question number 8 is from the chapter Neural Control and Coordination, and the question is receptor sites for neurotransmitters are present on. Options are presynaptic membrane, tips of axons, postsynaptic membrane, and membrane of synaptic vesicles. Okay, so what are neurotransmitters? These are the chemicals that help to transfer the nerve signal from one neuron to other. Okay, so let's write it down here. These are the chemical that help to transfer the nerve signal from one neuron to the other. Okay, one neuron to the other during the chemical synapse. Okay, and where are they stored? They are stored in the synaptic vesicles. So let's see. Suppose this is the neuron. Okay, these are the dendrites. Here we have the axon. Now at the terminal end of the axons we have synaptic knobs. Okay. Now if you look at the magnified image of this synaptic knob, here you will see there are synaptic vesicles. Okay. So these are synaptic vesicles which contain the neurotransmitters. Okay. So the neurotransmitters are stored in these synaptic vesicles. Okay. So synaptic vesicles containing neurotransmitters. Okay, here we have the postsynaptic neuron. Okay, this is the postsynaptic neuron. Postsynaptic. And here you will see there are receptor sites for these neurotransmitters. Okay, this is the cleft, synaptic cleft, that is the gap here. Okay, these neurotransmitters will be released from here. They will travel across this synaptic cleft and then they will come in contact with these receptors present on the postsynaptic neuron. Okay, so the correct answer will be option C, postsynaptic membrane. Okay. Question number 9 is again from the chapter Sexual Reproduction in Flowering Plants, and the question is coconut fruit is a. Options are berry, nut, capsule and drupe, and the correct answer is option D, that is drupe. Okay, so what is a drupe? Let's see. We know that in a fruit the outermost layer is epicarp, then comes mesocarp, and then the innermost layer is endocarp that encloses the seeds, right? Now, in a drupe type of fruit, the mesocarp is fibrous in nature. Okay, so there is a fibrous mesocarp and the endocarp is stony in nature, or we can say there is a hardened endocarp. Right, so this is the characteristic of a drupe. Right, now the most common examples of drupe are mango and coconut. In a mango, as you know, that suppose this is the mango, then there is a outermost layer, that is epicarp here, that we take out from the mango. Okay, then inside that there is edible portion that is fibrous in nature, and that is the mesocarp. Okay, so mesocarp is edible in a mango. Okay, and inside that there is stony endocarp. Okay, stony endocarp. Inside this stony endocarp, seeds are present. Got it? So this is simple. But for coconut, it is quite confusing, because in coconut, the edible part is not the mesocarp, it is the endosperm. Okay. Here the mesocarp is fibrous in nature, that looks hairy. Okay. So the coconut that we buy from the market, in that coconut, generally the outermost epicarp is not present, because that is already removed before we buy it from the market. Okay, so what we see as hairy structure, okay, hairy portion on the coconut, that is the mesocarp. Okay, that is the fibrous mesocarp. So here the mesocarp is not edible, and it is used to make the coir of the coconut. Okay, coir. Now inside that there is stony endocarp, and inside the endocarp there is endosperm that is edible in nature. Okay, so the only difference is that in mango the mesocarp is edible, but in coconut the mesocarp is not edible. Here the edible part is endosperm. Okay, but the common thing is that both have fibrous mesocarp, and inside that there is stony endocarp. Okay, so these two are drupe, so option D will be the correct answer. Now question number 10 is from body fluids and circulation, and the question is adult human RBCs are enucleated. Which of the following statements is or are most appropriate explanation for this feature? Okay, so let's see the statement. The first statement is that they do not need to reproduce. Yes, they do not need to reproduce, but the reason is that they are constantly being synthesized in the bone marrow. Okay, so the RBCs are synthesized in which part of the body? In the bone marrow. Okay, in bone marrow. What is the lifespan of RBCs? 120 days. So after 120 days, the RBCs are degenerated and new RBCs are constantly being synthesized in the bone marrow. That is the reason they do not need to reproduce. Okay, so this is not a proper explanation why they are enucleated. Enucleated means what? That during the maturation process, the nucleus is removed. Okay, so we know that the RBCs do not contain nucleus. Okay, so no nucleus is present. So actually the nucleus is removed from the RBCs during the maturation process. Okay. So the first statement is not the correct explanation of this. That means why the nucleus is removed. Then the next one is they are somatic cells. So yes, all the cells that are not germ cells are known as somatic cells. So these RBCs are also somatic cells. But again, this does not explain why the nucleus is removed from the RBCs. Okay. Then the third one is they do not metabolize. Yes, they do not metabolize. But the reason why they do not metabolize is that they do not contain any organelle. Okay. So no organelles are present. Therefore, they do not show any metabolic activity. Okay. And the last statement says all their internal space is available for oxygen transport. Yes, this is the most appropriate explanation why the nucleus is removed. Okay, so when the nucleus is removed from the RBCs, it creates more space. More space is created. More space created for oxygen transport. Okay, and we know that the most important function of RBC is the transport of oxygen in the body. Okay, so D will be the most appropriate explanation. So option D, only D will be the correct answer. Okay. Question number 11 is from the chapter Human Reproduction, and the question is capacitation occurs in. Options are epididymis, vas deferens, female reproductive tract and reti testis. So what is capacitation? It is a process in which the sperm is activated for fertilization. Okay, so let's write it down here. It is a biochemical process which enables the sperm to fertilize an ovum. To fertilize an ovum. Okay. And where does fertilization take place? It takes place in the fallopian tube. We all know that. Fallopian tube, which is part of female reproductive tract. Right? Female reproductive tract. So there are certain secretions in the female reproductive tract which enables this process, that is capacitation. Okay, so option C will be the correct answer. Other options, that is epididymis, vas deferens and reti testis, are part of male reproductive system. Right. Question number 12 is from the chapter Biological Classification, and the question is which of the following are found in extreme saline conditions? Options are eubacteria, cyanobacteria, mycobacteria and archaebacteria. Okay, so we know that all the bacteria that are able to survive in extreme conditions are examples of archaebacteria. Okay, it is very simple. So here extreme saline condition is mentioned. That means we are talking about the halophiles. Okay. But other than halophiles, there are certain bacteria which are able to survive in the hot springs. Hot springs. So this is also extreme condition. Okay. These bacteria are known as thermoacidophiles. Thermoacidophiles. Similarly, the bacteria that live in the marshy areas are called methanogens. We know that methanogens live in the gut of ruminant animals, right? So all these halophiles, thermoacidophiles and methanogens, these are examples of archaebacteria. That means primitive type of bacteria. Okay, primitive type of bacteria. These are different from eubacteria because they have a different cell wall structure. Different cell wall structure that enables them to survive in extreme conditions. Okay, so option D will be the correct answer. Question number 13 is from the chapter Organisms and Population, and the question is asymptote in a logistic growth curve is obtained when? Okay, so asymptote in a logistic growth curve is represented by the straight line that is associated with the logistic growth curve. Okay, that means if I draw the logistic growth curve, here we have the population size, this side we have time. Okay, so initially we know that there are plenty of resources, so the population utilize that resource and increases at a fast rate. Okay, so the population is accelerated, but later on the resources are being utilized continuously, so there is a decrease in resources and then the population also starts decreasing. Okay, and after some time it becomes a straight line here. Okay, in this curve you will get a straight line. Okay, so at this point it is said that the population has become equal to the carrying capacity. Okay, so this part is the asymptote, right? So which option will be the correct one? Option number A, K is equal to N. Okay. Question number 14 is from the chapter Evolution, and the question is artificial selection to obtain cows yielding higher milk output represents option A, directional as it pushes the mean of the character in one direction. Option B, disruptive as it splits the population into two, one yielding higher output and the other lower output. Option C, stabilizing followed by disruptive as it stabilizes the population to produce higher yielding cows. And the last one is stabilizing selection as it stabilizes this character in the population. Okay, so we know that in case of natural selection, that means selection done by the nature, there are three different types of selections. First one is stabilizing selection. Stabilizing selection. Second one is directional selection. Directional. And the last one is disruptive selection. Okay, disruptive selection. Now let's draw the graph for each one of them. The graph for stabilizing selection will look like this. This is the initial curve and this is one curve. That means there is only one mean value. Only one mean value is there. Okay, and in the second one, that is directional, the graph will look like this. This is the mean value and later on it will shift in one direction. Okay, so there will be one mean value but the direction of the mean value will change. Okay, direction of mean value changes. And in case of disruptive, the graph will look like this. In this case there are two mean values. Okay, two mean values are there. Okay, now what is the difference? In stabilizing selection, the selection is done against two extreme traits. Okay, if there are two different traits, for example, low milk output and high milk output. Okay, these two are two extreme traits. So in case of stabilizing selection, there will be something intermediate. Okay, so it will be against extreme value. Okay, so the selection is done against two extreme of the traits. But here in this question, we are not stabilizing the character. Okay, here we are choosing one extreme character, that means high milk output. Okay, so this will be an example of directional selection. What happens in directional selection? The selection is done such that one extreme trait is selected. Okay, one extreme trait is selected. So as we have seen that out of the two extreme traits, we are selecting one extreme trait, that is high milk output. Okay, disruptive selection, what is this? Here two mean values are obtained, that means two characters are present. Okay, two characters are present. But in this question, we are talking about only one character, that is milk output. Okay, so that will be an example of directional selection. That means which option will be the correct one? First option, directional, as it pushes the mean of the character in one direction. Okay, so option A is the correct answer. Question number 15 is from the chapter Mineral Nutrition, and the question is select the mismatch. So in the first one, Rhodospirillum is matched with Mycorrhiza. Now we know that Rhodospirillum is a facultative anaerobe. Okay, so this is an anaerobic bacteria. Okay, anaerobic bacteria, and this is also a nitrogen fixer. Nitrogen fixer. That means it fixes nitrogen. Okay, it converts the atmospheric nitrogen into ammonia. Okay, now the mycorrhiza is the symbiotic association of roots of higher plants, that means roots of vascular plants, higher plants, and fungi. Okay. So in mycorrhiza, fungi are involved, not the bacteria. Okay. Therefore, this is an incorrect match. Now in the second one, Anabaena is matched with nitrogen fixer. Yes, this is correct because Anabaena is a bacteria which is a nitrogen fixer. Okay. Third one is Rhizobium and it is matched with alfalfa. So Rhizobium is also a bacteria. Okay. Rhizobium is also a nitrogen fixing bacteria. And it remains in symbiotic association with the roots of leguminous plants. Okay. Symbiotic association with the root of leguminous plants such as garden pea, alfalfa, etc. Okay. Therefore, alfalfa is a correct match for Rhizobium. So option C is also correct. Now the last one is Frankia and it is matched with Alnus. So Frankia is a gram-positive species of bacteria. Okay. Gram-positive of actinomycete filamentous bacterium that lives in symbiotic association with the plants of genus Alnus. Okay. So Alnus is the genus of the plants. And this is also a correct match. Okay. So the only mismatch is the first one, that is option A. So this will be the correct answer. Question number 16 is from the chapter Neural Control and Coordination. And the question is, good vision depends on adequate intake of carotene-rich food. Select the best option from the following statements. So the first statement says that vitamin A derivatives are formed from carotene. So this is correct. Okay. Carotene synthesize vitamin A derivatives. So this is a correct statement. Now the second statement says the photopigments are embedded in the membrane disk of the inner segments. So this is incorrect. We know that retina has two segments, inner segments and outer segments, right? Inner segment and outer segment. Now this outer segment contains the photopigments, that is rods and cones. Okay. So rods and cones are the photopigments that are present in the outer segment of the retina. Okay. So this is incorrect statement. Now the third one says that retinal is a derivative of vitamin A. This is also a correct statement. Okay. So carotene synthesize vitamin A derivatives. That means retinal. Okay. Retinal is the most important vitamin A derivative for our vision. Okay. So what is retinal? It is a light sensitive pigment. Okay. Light sensitive pigment. Now this retinal binds with the opsin protein. Okay. Opsin to form rhodopsin. Rhodopsin, that is a light absorbing molecule. Light absorbing molecule. Okay. Now let's come to the last statement. It says that retinal is a light absorbing part of all the visual photopigments. So this we have already discussed. Okay. So option D is also correct. So how many statements are correct here? Statement A, statement C and statement D are correct. Okay. So option A will be the correct answer. Question number 17 is from the chapter Biotechnology Principles and Processes. And the question is, the DNA fragments separated on an agarose gel can be visualized after staining with. Options are acetocarmine, aniline blue, ethidium bromide and bromophenol blue. So here we are talking about the agarose gel electrophoresis, right? Agarose gel electrophoresis. So this is a technique which is used for the separation of DNA fragments. Okay, separation of DNA fragments under the effect of electric fields. Okay, so after this is done, the DNA fragments can be visualized after staining it with a compound that is known as ethidium bromide. Okay, staining with ethidium bromide, because the pure DNA cannot be seen by the naked eyes. Okay, and after staining with ethidium bromide, then it needs to be exposed to the UV light of the sun. Okay, so the next step is exposure to UV light, and then bright orange colored bands of DNA fragment can be seen. Okay, bright orange bands of DNA are seen. Okay, so here it was asked that which compound is used for staining. So it will be ethidium bromide, that is option C. Okay. Question number 18 is from the chapter Body Fluids and Circulation, and the question is, the hepatic portal vein drains blood to liver from. Options are stomach, kidneys, intestine and heart. So have you heard of hepatic portal system? Hepatic portal system. It is a unique vascular connection between digestive tract and the liver. Okay, so vascular connection between the digestive tract and liver. Okay, that means the blood will be carried between the digestive tract and the liver. Okay, so here in this system, the hepatic portal vein carries blood from intestine to the liver. Okay, carries blood from intestine to liver before it is delivered to the systemic circulation. Okay, so the correct answer will be option C, that is intestine. Okay, blood is carried from intestine to the liver. Okay. Question number 19 is from the chapter Anatomy of Flowering Plants, and the question is, the vascular cambium normally gives rise to. Options are primary phloem, secondary xylem, periderm and phloem. So we know that the vascular bundles consist of xylem and phloem, right? Xylem for the transport of water and minerals, and phloem for the transport of food. Okay, these are primary tissues. Okay, that means primary xylem and primary phloem. Now in dicot plants, there is an additional layer of tissue that is known as cambium in between the xylem and phloem. Okay, so suppose this is your xylem, upper portion is phloem. Okay, so the tissue found between these two is the cambium. Okay, in case of dicot plants. Now due to the presence of such cambium, these vascular bundles possess the ability for the formation of secondary tissues. Okay, so this cambium is responsible for the formation of secondary tissues. That means secondary xylem and secondary phloem. Okay, therefore these are also known as open vascular bundles. Okay, now in the options, the first one is primary phloem, so this will not be true because primary xylem and primary phloem are already present with or without cambium. Okay, so the cambium is responsible for the formation of secondary xylem and secondary phloem. So in the options, secondary xylem is mentioned, so option B will be the correct answer. Question number 20 is from the chapter Principles of Inheritance and Variation, and the question is, thalassemia and sickle cell anemia are caused due to a problem in globin molecule synthesis. Select the correct statement. Okay, so we know that these two diseases, thalassemia and sickle cell anemia, are related to globin molecule defect. Okay, defect in globin molecule, and we know that globin chain make up the hemoglobin, right? Hemoglobin. That means in both the diseases, hemoglobin will be affected. Okay. Now, let's talk about each one of them. If we talk about thalassemia, Basically, we'll see the difference between these two. Okay, so in thalassemia, the defect is due to mutation or deletion of a gene. Okay, so there is mutation or deletion of gene, and it results in reduced rate of synthesis of globin chain. Okay, reduced rate of synthesis of one of the globin chains. Okay, globin chains. So when the rate of synthesis will be reduced, that means less number of globin chains will be formed. Okay, less number of globin molecules are synthesized. Okay, and less number means this is an example of quantitative defect. Okay, quantitative defect, because it is affecting the quantity of the globin molecule. Okay, now if we talk about the sickle cell anemia, this is not a quantitative defect, instead it is a qualitative defect. Okay, qualitative defect, because it does not affect the number or the amount of globin molecules, it affects the function. Okay, so function of globin molecule is affected. Globin molecule is affected. Okay, what happens? Incorrect or abnormal functioning globin is formed. Okay, so the main difference is that thalassemia is a quantitative defect and sickle cell anemia is a qualitative defect. Okay, now let's see the options. In the first option, we have both are due to quantitative defect in globin chain synthesis. So this is incorrect, because we have seen that one is due to quantitative defect and the other is due to qualitative defect. Okay, now the next statement says thalassemia is due to less synthesis of globin molecule. Yes, okay, this is a quantitative defect and there is less synthesis of globin molecules. Okay, so this is correct statement. Now the third statement says sickle cell anemia is due to quantitative problem of globin molecules. Incorrect, because we have seen that sickle cell anemia is due to qualitative defect. Okay, abnormal functioning globin is formed. Okay, now the last statement says both are due to qualitative defect. Again incorrect, because one is due to qualitative and the other is due to quantitative defect. Okay, so the only correct statement is the second one. That means option B will be the correct answer. Question number 21 is from the chapter Body Fluids and Circulation, and the question is the genotypes of a husband and wife are IAIB and IA small I. Among the blood types of their children, how many different genotypes and phenotypes are possible? Okay, so let's see. One type of genotype is IAIB. Okay, and the wife has genotype IA and small I. Okay, so let's see. First one will be IAIA, second type will be IAIB, then IA small I and IB small I. Okay, so these four types of genotypes are present. Okay, these are four different types of genotypes. These are the genotypes. How many? Four genotypes. Okay, now if we talk about the phenotypes, so the first one is IAIA, that means the blood group will be A. Second one is IAIB, that means the blood group will be AB. Third is IA small I, that means the blood group will be A. And the last one is IB small I, that means the blood group will be B. Okay, so two A are present, that means this is one type of phenotype. Okay, then AB the second type and B the third type. So three types of phenotypes are present. Okay, so four genotypes and three phenotypes. Okay, that means option B will be the correct answer. Okay, now question number 22 is from the chapter Transport in Plants, and the question is which of the following facilitates opening of stomatal aperture? Okay, so let's see. Suppose these are guard cells. We know that guard cells are the kidney shaped cells, right? So these are guard cells and this is the stomatal pore or stomatal aperture. Stomatal aperture. So right now the stomatal aperture is closed. It will open when water will enter the guard cells. Okay, so when water enters the guard cells, when water enters the guard cells, what happens? The turgidity increases. Turgidity increases. And when turgidity increases, the outer wall of the guard cells bulges outwards like this, whereas the inner wall of the guard cell moves inwards. Okay. So this leads to the opening of the stomatal aperture, right? So this is the opened stomatal aperture. Now, this movement of the guard cells is due to the cellulose microfibrils, okay, and more specifically due to the radial orientation of cellulose microfibrils that are present in the cell wall of the guard cells. Okay, so radial means in this direction and longitudinal means in this direction. Okay, so here you can see that there is change in the radial orientation. Okay, so the correct answer will be option number B, that is radial orientation of cellulose microfibrils in the cell wall of the guard cells. Okay, so option B is correct answer. Question number 23 is from the chapter Morphology of Flowering Plants, and the question is, in Bougainvillea, thorns are modifications of. Options are adventitious root, stem, leaf and stipules. So this is very simple. Okay, we know that in Bougainvillea, thorns are the modification of stems, okay, in which the axillary buds of the stems get modified into the thorns. Okay, so let's write it down here. Axillary buds of stems get modified into woody, straight and pointed thorns. Okay, straight and pointed thorns. So option B will be the correct answer. Question number 24 is from the chapter Biodiversity and Conservation, and the question is, which one of the following is related to ex situ conservation of threatened animals and plants? So what is ex situ conservation? It means the method of conservation in which the animals are taken care of outside their natural habitat. Okay, outside their natural habitat. Now the first one, that is biodiversity hotspots, are the natural habitat of the animals. Okay. Second one, Amazon rainforest is also natural habitat, and the third one, that is Himalayan region. So all these three are the natural habitats of animals and plants. Natural habitats. Whereas wildlife safari parks is an ex situ method. Ex situ method in which the animals are allowed to roam free. Okay, so this is a park like zoo, but in normal zoo you see that the animals are kept in confined places, but in these wildlife safari parks the animals are allowed to roam free. Okay, in which animals are allowed to roam free, and this is done to give them the experience of their natural habitat. Okay, to resemble their natural habitat. And the visitors can observe them by riding through the park in vehicles. Okay, so the correct answer will be option number D, wildlife safari parks. Question number 25 is from the chapter Morphology of Flowering Plants, and the question is, root hairs develop from the region of. Options are elongation, root cap, meristematic activity and maturation. So let's see the regions of the root tip. Here we have root cap. Okay, this is the root cap, and just few millimeters above the root cap there is a region of meristematic activity. Okay, so this is the region of meristematic activity. In this region, what happens? The cells undergo repeated division. So repeated cell division takes place in this region. Okay. Now above this region, there is a region of elongation. So this is the region of elongation. Here the cells undergo elongation and enlargement, and this region is responsible for the increase in length of the root. Okay, increase in root length. And finally, the top most region is the region of maturation. So this is the region of maturation. This region, the epidermal cells of this region gives rise to the root hairs. Okay. So these are the root hairs. So the question is asked that these root hairs develop from which region? So region of maturation. That means option D will be the correct answer. Question number 26 is from the chapter Principles of Inheritance and Variation, and the question is, a disease caused by an autosomal primary non-disjunction is. Options are Klinefelter syndrome, Turner syndrome, sickle cell anemia and Down syndrome. Okay, so what do we mean by non-disjunction? Non-disjunction. It means the failure of the chromosomes to separate during the cell division. Okay, so let's write it down here. Failure of chromosomes to separate. Now if the chromosomes do not separate, what will happen? It will result in abnormal number of chromosomes. Okay, so it results in abnormal number of chromosomes. That means the number of chromosomes will be more than required. Okay, now let's check. The first option is Klinefelter syndrome. Yes, this involves non-disjunction, but here sex chromosomes are involved. Okay, so it involves non-disjunction of sex chromosomes, and the resulting karyotype in this is XXY. So as you can see, there is one additional X chromosome present here. Okay, X and Y are sex chromosomes. So this will not be the correct answer because the question is asked about autosomal primary non-disjunction. That means there must be the non-disjunction of autosomal chromosomes, not the sex chromosomes. Okay. Now the second one is Turner syndrome. So Turner syndrome is caused due to absence of one X chromosome in females. Okay. So this is due to the absence of chromosome, not the inability of chromosome to separate. So this will not be the correct answer. And the third one is sickle cell anemia. We know that sickle cell anemia is an example of point mutation. Okay, point mutation, where the amino acid, that is glutamic acid is substituted by valine at 6th position of the beta globin chain. Okay, so this is not related to the non-disjunction. Now the last one is Down syndrome. So yes, it involves non-disjunction of autosomal chromosomes. Okay, autosomal chromosomes. We know that it results in the trisomy of chromosome number 21. So this will be the correct answer. Option D will be the correct answer. Okay. Question number 27 is from the chapter Transport in Plants, and the question is, the water potential of pure water is. Options are less than 0, more than 0 but less than 1, more than 1, and the last option is 0. Okay, so the correct answer is option D, that is 0. Okay, so water potential is expressed in the unit of pressures, okay, such as pascals. Okay, so unit is taken as that of pressure. For example, Pascal, and the symbol for water potential is a Greek symbol known as psi. Okay, and it is written like this. Right, now it is assumed by convention that the water potential of pure water at standard temperature which is not under any pressure is 0. Okay, so option D, 0 will be the correct answer. Question number 28 is from the chapter Cell Cycle and Cell Division, and the question is, which of the following options gives the correct sequence of events during mitosis? So we know that during mitosis there are 4 stages. First one is the prophase, second one is metaphase, third is anaphase, and the last one is telophase, right? Now, what happens in the first stage, that is prophase? In this, the condensation of chromosomal material takes place. Condensation of chromosomal material to form compact mitotic chromosomes. And other than that, there is one more important event of prophase, that is disassembly of the nuclear membrane. Disassembly of nuclear membrane. Now the next is metaphase. So the most important event of metaphase is arrangement of the chromosomes at the equator. Arrangement of chromosomes at equator through the spindle fibers. Through spindle fibers. Next one is anaphase. So in this phase, the centromeres split and the chromatids separate. Okay, so splitting of centromere and separation of chromatids. Separation of chromatids. That means segregation takes place. Segregation takes place during anaphase. Okay, and these separated chromatids will start moving towards the opposite pole. Okay, move toward opposite poles. Now, in telophase, the chromosomes will cluster at opposite poles. So, cluster of chromosomes at opposite poles. And we have seen that during the prophase, the nuclear membrane disassembly had taken place. But in this last stage, that is telophase, the nuclear envelope will redevelop. Okay, so development of nuclear envelope. Also, nucleolus, Golgi bodies and endoplasmic reticulum will be reformed here. Okay. Now let's see. First one is condensation of chromosomal material, then disassembly of nuclear membrane. So in the first option we have condensation, then nuclear membrane disassembly. This is correct. Then metaphase, that means arrangement of chromosomes at equator. This is also correct. Then centromeres division, that means anaphase, right? Anaphase. So anaphase involves centromere division as well as segregation. Okay, and then last one is telophase. So this is the correct sequence, right? Now let's check others. The second one is condensation, then crossing over. So crossing over is involved in the meiosis. Okay, during meiosis we see the crossing over, not during mitosis. The third one is condensation, then arrangement at equator. So condensation, that means prophase, and arrangement at equator, that means metaphase. But before arrangement at equator, the disassembly of nuclear membrane also takes place. So the first one is better than this option. Okay, now let's check the last one, condensation, the nuclear membrane disassembly. This is correct. Then again crossing over, which is part of meiosis. So the only correct option is the first one. Option A will be the correct answer. Question number 29 is from the chapter Biotechnology Principles and Processes, and the question is, the process of separation and purification of expressed protein before marketing is called. Options are downstream processing, bioprocessing, post-production processing and upstream processing. So this is a direct question. The correct answer will be downstream processing. Okay, so after the completion of biosynthetic stage of certain product, the product has to be subjected through a series of process in order to make it ready for marketing. Okay, so the processes involved are collectively known as downstream processing, and this process involves separation and purification of the expressed protein. Okay, so option A will be the correct answer. Question number 30 is from the chapter Human Reproduction, and the question is, a temporary endocrine gland in the human body is, options are corpus caardiacum, corpus luteum, corpus allatum and pineal gland. So it is clear that option A and option C cannot be the correct answer because these are present in insects, not in humans. Okay. Now, out of option B and option D, we know that pineal gland is a permanent gland, not a temporary gland. Okay, permanent gland, and it is very important gland of our body, and this is present in the brain. But the question is asked about the temporary endocrine gland, so the correct answer will be corpus luteum. We know that it is formed after ovulation, during the luteal phase. Okay, during luteal phase, the corpus luteum is responsible for the secretion of progesterone. Okay, and since it is confined to only one specific phase of the menstrual cycle, that is luteal phase, so this is a temporary endocrine gland, not a permanent gland. So option B will be the correct answer. Question number 31 is from the chapter Anatomy of Flowering Plants, and the question is, which of the following is made up of dead cells? Options are collenchyma, phloem, phloem and xylem parenchyma. So the correct answer is phloem. Okay, phloem is made up of dead cells. Actually it is suberized cork cells. Okay, that means it has deposition of suberin. Okay, suberized cork cells in which the cork cambium, that is also known as phelogen, cut towards the outside. Okay, cut towards the outside. If we talk about collenchyma, so we know that collenchyma is a supporting tissue. It is a supporting tissue. Okay, and it is made up of what kind of cells? Elongated living cells. Okay, it is made up of elongated living cells with unevenly thickened. Okay, so there is thickening in an uneven manner. Okay, and non-lignified cell walls. Non-lignified cell wall. Let's come to the third option, that is phloem. So this is very simple. We know that phloem is the food conducting element, right? Food conducting element. That means it helps in the transportation of food. Then the last one is xylem parenchyma. So xylem parenchyma is used to store water. And we know that water is very essential for the functioning of the plant. So these three, option A, option C and option D, these are made up of living cells, whereas option B, that is phloem, is made up of dead cells. Therefore option B will be the correct answer. Question number 32 is from the chapter Plant Kingdom, and the question is, an example of colonial alga is, options are Volvox, Eulothrix, Spirogyra and Chlorella. So the correct option is option number A, that is Volvox. Okay, so this is the colonial forms. That means many cells are grouped together to form a colony. Okay, together to form a colony. If we talk about the second and third option, that is Eulothrix and Spirogyra, these two are filamentous forms. Whereas Chlorella is the unicellular form. Unicellular, that means only one cell. Unicellular form. Other than Chlorella, Chlamydomonas is also example of unicellular form. Okay, Chlamydomonas. Okay. So according to the question, the correct answer is option number A, Volvox. Question number 33 is from the chapter Reproductive Health, and the question is match the following sexually transmitted diseases in column 1 with their causative agent in column 2 and select the correct option. Okay, so in the first column we have different sexually transmitted diseases. The first one is gonorrhea. Now it is a bacterial disease and it is caused by a bacterium named Neisseria gonorrhoeae. Okay, this second one, Neisseria gonorrhoeae. So it affects the mucous membrane of the reproductive tract. Okay, mucous membrane of the reproductive tract. So the correct match for A is number 2. That means the last option. Okay, here A is matched with 2, but let's see other diseases also. The second one is syphilis. So syphilis is again a bacterial disease and it is caused by a bacterium Treponema pallidum. Okay, this third one, Treponema pallidum. Okay, so the correct match for B will be number 3. Yes, this is also matched here in the last option. Then the third one is genital warts. Genital warts is a viral disease. Okay, this is caused by a virus and the name of the virus is human papilloma virus, that is HPV. Okay, and it is passed on through the direct skin to skin contact with the affected person. Okay, so the correct match for C will be number 4. This is also correctly matched in option number D. And the last one is AIDS. So this is very common disease. This is also a viral disease and it is caused by the human immunodeficiency virus HIV, that is number 1. Okay, so we know that it affects the immune system of our body. Okay, immune system is affected. So the correct match for A is D is number 1. Okay, this one. So option D will be the correct answer. Question number 34 is again from the chapter Reproductive Health, and the question is the function of copper ions in copper releasing IUDs is option A, they inhibit gametogenesis, option B, they make uterus unsuitable for implantation, option C, they inhibit ovulation, and the last one is they suppress the sperm motility and fertilizing capacity of sperms. Yes, this is the correct answer. Okay, what are the examples of copper releasing IUDs? The examples are copper T, copper 7 and multi-load 375. Okay, multi-load 375, these are the examples mentioned in the NCERT. Okay, so the role of copper ions here is to suppress the sperm motility and fertilizing capacity of the sperms, and overall these copper releasing IUDs, what do? They increase the phagocytosis. Okay, these IUDs increase the phagocytosis of sperms. Phagocytosis of sperms within the uterus. Okay, so according to the question, the correct answer is option number D. Question number 35 is from the chapter Microbes in Human Welfare, and the question is which of the following in sewage treatment removes suspended solids? Options are secondary treatment, primary treatment, sludge treatment and tertiary treatment. So the correct answer is primary treatment. This is also known as physical treatment because here the physical methods are used. Okay, physical methods are used and it basically involves sequential filtration and sedimentation. Okay, sequential filtration and sedimentation. Now if we talk about the secondary treatment, it is known as biological means or biological method. Okay, biological method, because here the microorganisms are used. Okay, so this process involves oxidation of organic matter by the aerobic microorganisms. Okay, organic matter by aerobic microorganisms such as bacteria and fungi. Okay, sludge treatment, we know that the effluent that is formed is passed to a settling tank where bacterial flocks are allowed to sediment. Okay, so those bacterial flocks are known as activated sludge. Okay, bacterial flocks are known as activated sludge. Now it needs to be treated. So what is done? The maximum amount of this activated sludge is pumped into large tanks known as anaerobic sludge digester. Okay, anaerobic sludge digester. Here, some other types of bacteria known as anaerobic bacteria digest these aerobic bacteria. Okay. Now the tertiary treatment, here the extra filtration takes place. Extra filtration. That means some leftover contaminants are removed in this process. Okay. Leftover contaminants are removed. But here the question was asked about the removal of suspended solids. So it takes place in the primary treatment. So option B will be the correct answer. Okay. Question number 36 is from the chapter Animal Kingdom, and the question is, an important characteristic that hemichordates share with chordates is. Okay. So what are hemichordates? These are the connecting link between echinoderms and chordates. Okay. Now, let's see. The first option is ventral tubular nerve cord. So what is nerve cord? It is a cord or the nervous tissue that is present above the notochord. Okay. Now, it is common in hemichordates and chordates, but it is not ventral. It is dorsal. Okay. It is dorsal. And in hemichordates, it is restricted to the collar region. Okay. Collar region. So it is not ventral, it is dorsal. So option A will not be the correct answer. Second one is pharynx with gill slits. Yes, this is common in hemichordates and chordates. Okay, pharyngeal gill slits are found in both of them. So this can be the correct answer. Let's see the third one. Pharynx without gill slits. This is incorrect because gill slits are present. Okay. Then the last one is absence of notochord. This is again incorrect because notochord is present in chordates. Okay, it is present in chordates and it is tubular and dorsal. Okay, whereas it is absent in hemichordate. It is absent in hemichordate. So this is the presence or absence of notochord is not common between them. The only common thing is pharyngeal gill slits. Therefore option B will be the correct answer. Question number 37 is from the chapter Molecular Basis of Inheritance, and the question is, the final proof for DNA as the genetic material came from the experiments of. So this is a very easy and direct question. We know that the unequivocal proof of DNA being the genetic material was given by the scientists Hershey and Chase. Okay, so they proved that DNA is the genetic material using the bacteriophages. Okay, so bacteriophages were used in their experiments. And they showed that these bacteriophages inject their genetic material into the host cell, leaving the protein coat outside the cell. Okay, so the correct answer will be option number A, Hershey and Chase. Question number 38 is from the chapter Principles of Inheritance and Variation, and the question is, among the following characters, which one was not considered by Mendel in his experiments on peas? Okay, so we know that Mendel performed his experiment on the garden peas that is known as Pisum sativum. Okay, scientific name is Pisum sativum. So he considered different characteristics. So let's see. The first option is trichomes, glandular or non-glandular. So this is not correct. Okay, the trichomes was not considered in his experiments. Next one is seed, green or yellow. Yes, seed color was considered. So this is correct. This is not correct answer, but this is considered in the Mendel's experiment. Okay. Then the third one is pod, inflated or constricted. Yes, so pod shape was also considered. Then stem size, tall or dwarf was also considered. So these three were considered, but the first one was not considered. Therefore, this will be the correct answer. Okay. Question number 39 is from the chapter Morphology of Flowering Plants, and the question is, plants which produce characteristics pneumatophores and show vivipary belongs to. Options are halophytes, samophytes, hydrophytes and mesophytes. Okay. So the correct answer will be option number A, that is halophytes. Okay, these are the plants that grow in, salt rich soil. Salt rich soil. So they are able to adapt in the high salinity conditions. In these plants, the roots are modified into pneumatophores. Okay, so pneumatophores are the modified form of roots. Modified roots which grow outward. So normally the roots grow inside the soil, but in this case they grow outwards. Grow outwards in order to obtain oxygen. Okay, to obtain oxygen. And they show vivipary, that means seed germination takes place while still attached to the parent plant. Okay, and the example of halophytes is mangroves. Mangroves. Okay, so the correct answer will be option number A. Question number 40 is from the chapter Locomotion and Movement, and the question is, the pivot joint between atlas and axis is a type of. Options are cartilaginous joint, synovial joint, saddle joint and fibrous joint. Okay, so here the correct answer will be option B, that is synovial joint, because these are the joints which allow considerable movement. Okay, considerable movement. And the most important characteristic of synovial joint is the presence of fluid-filled synovial cavity. Fluid-filled synovial cavity between the articulating surfaces of two bones. Okay, so this pivot joint is an example of synovial joint. Now let's see other options. The first one is cartilaginous joint. So in this type of joint, the bones are joined with the help of cartilages, and this type of joint allow only limited movement. Okay, unlike the synovial joints which allow considerable movement, these cartilaginous joints allow only limited movement. The example of cartilaginous joint is the joint present between the adjacent vertebra. Okay, adjacent vertebra or vertebrae in the vertebral column. Vertebral column. Third option is saddle joint. So saddle joint is found between the carpals and metacarpals. Okay, carpals and metacarpals. And this is also an example of synovial joint. Okay, because considerable movement is possible here. Okay, now the last one is fibrous joint. In this type of joint, no movement is possible. Okay, they do not allow any movement, and these are found in the skull. Okay, these skull bones, the flat skull bones, are attached with the help of fibrous joint. So here the correct answer will be option B, synovial joint. Question number 41 is from the chapter photosynthesis, and the question is, with reference to factors affecting the rate of photosynthesis, which of the following statements is not correct? Okay, incorrect statement has to be identified. Let's see the first statement. It says that increasing atmospheric carbon dioxide concentration up to 0.05% can enhance carbon dioxide fixation rate. This is absolutely correct statement. Okay. Then the second statement says C3 plants respond to higher temperatures with enhanced photosynthesis, while C4 plants have much lower temperature optimum. So this is not correct, because the photosynthetic activity of the C3 and C4 plants is mainly affected by the enzymes and not by the temperatures. Okay, so let's write it down here. Photosynthesis is affected by the enzymes. Affected by enzymes and not by the temperature. If we talk about the C3 plants, C3 plants have what enzyme? Rubisco enzyme. Okay, Rubisco enzyme, which gets activated on higher carbon dioxide concentration, and C4 plants have PEP, phosphoenol pyruvate, as the major carbon dioxide acceptor. So these enzymes are responsible for the rate of photosynthesis, not the temperatures. Okay. In fact, higher temperature can lead to destruction of photosynthetic pigments, so it will not enhance the photosynthesis in C3 plants. So this is absolutely incorrect statement. Okay. Now let's come to the third statement. It says that tomato is a greenhouse crop which can be grown in carbon dioxide enriched atmosphere for higher yield. This is also a correct statement. Okay. And the last statement says that light saturation for carbon dioxide fixation occurs at 10% of the full sunlight. This is also correct statement. So these things are mentioned in NCERT. Okay. Option B will be the correct answer because this is not correct statement. Okay. Question number 42 is from the chapter Molecular Basis of Inheritance, and the question is DNA fragments are, options are negatively charged, neutral, either positively or negatively charged depending on their size, and the last one is positively charged. So this is a direct question. We know that DNA are always negatively charged. Okay, therefore these wrap around the positively charged protein molecules. Okay, therefore option A will be the correct answer. Question number 43 is from the chapter Biological Classification, and the question is which of the following components provides sticky character to the bacterial cell? Options are nuclear membrane, plasma membrane, glycocalyx and cell wall. So it is also a simple one. The answer will be glycocalyx. Okay. So the sticky nature is due to the glycocalyx, that is the slime layer rich in glycoprotein. Okay. Slime layer rich in glycoprotein. These glycocalyx can take the shape of loose sheath. Okay, the shape of loose sheath or a thick stiff mucilage coating. Okay, stiff mucilage coating that is known as capsule. Okay, so the correct answer will be option number C. Question number 44 is from the chapter Digestion and Absorption, and the question is which of the following options best represents the enzyme composition of pancreatic juice? So let's see. The first option is amylase, pepsine, trypsinogen and maltase. So we know that maltase is found in Succus entericus. Okay, Succus entericus. That means intestinal juice, not the pancreatic juice. So option A is incorrect. Then the next one is peptidase, amylase, pepsine and renin. Again we know that renin is a proteolytic enzyme that is found in the gastric juice of infants. Gastric juice of infants, because it helps in the digestion of milk proteins. Okay, so this one is also incorrect. Then the next one is lipase, amylase, trypsinogen and procarboxypeptidase. Yes, all these are found in the pancreatic juice. So this is the correct answer. Now let's see the last one. It is amylase, peptidase, trypsinogen and renin again. So we have already seen renin is found in gastric juice of infants, not in the pancreatic juice. So the correct option will be option number C. Question number 45 is from the chapter Animal Kingdom, and the question is which among these is the correct combination of aquatic mammals? Okay, so mammals means the animals that give birth to young ones. Okay, give birth to young ones, and they show the characteristic of lactation as well. That means they have the mammary gland, so they provide milk to the young ones. Now let's see, the first option is dolphins, seals and trigon. So dolphins and seals are mammals, but trigon are not mammals. Okay, trigon are also known as stingray, and these are cartilaginous fishes. Okay, cartilaginous fishes, not mammals. Now the next one is whales, dolphins and seals. Yes, all these three are mammals. So this can be the correct answer. Third one is trigon. Again, it is not a mammal, we have seen. So this is also incorrect. Then the last one is seals, dolphins and sharks. So seals and dolphins are mammals, but sharks are not mammals. These are also cartilaginous fishes. Cartilaginous fish. So the correct option is option number B. Question number 46 is from the chapter Plant Growth and Development, and the question is fruit and leaf drop at early stages can be prevented by the application of, options are ethylene, auxins, gibberellic acid and cytokinins. So the correct answer is option B, that is auxins. Okay, so we know that auxins are produced by the tips of root and shoot, right? So let's write it down here. produced by root and shoot tips. And what is the most important characteristic of auxins? The most important characteristic is apical dominance. Apical dominance, you must be knowing this. Another important characteristic is that it prevents the fruit and leaf drop at early stages as mentioned in the question, but at the same time it accelerates the fall of mature and older leaves and fruits. Okay. If we talk about other options, ethylene is a gaseous hormone which has the most important role being fruit ripening. Okay. So fruit ripening is the most important characteristic of ethylene hormone. Most important role of this hormone. Okay, fruit ripening, then flower wilting, and it regulates both growth as well as senescence. Okay, so let's write it down. It regulates both growth and senescence. Now the third one is gibberellic acid. So what is the most important role of gibberellic acid? It is cell elongation. Okay, cell elongation. It helps in the elongation of the stems, or overall it helps the plant to grow taller. Okay, so helps the plant to grow taller. And there is one more important characteristic of gibberellic acid, that is seed germination. Okay, seed germination. It helps in seed germination. Then let's come to the last one, that is cytokinins. It has the most important role, that is cell division. Okay. So we have seen the role of each of the hormones mentioned here. The correct answer will be auxins. Okay. Option number B. Question number 47 is from the chapter Structural Organization in Animals, and the question is, select the correct route for the passage of sperms in male frogs. Okay, so we know that in male frogs the sperms are produced in the testis. Okay, in the testis. Then the mature sperms are transported from testis into the vasa efferentia. Vasa efferentia. The vasa efferentia then enters the kidney. Okay, it enters the kidney after passing through the mesorchium. Okay, now after it enters the kidney, then the sperms enter the Bidder's canal from here. Okay, from kidney they move to the Bidder's canal. After that they enter the urogenital duct. Urogenital duct, and finally they exit through the cloaca into the water. Okay, cloaca into the water, and when they move to the water they fertilize the eggs which are released in water by the female frogs. Okay, so this will be the correct sequence. First there will be testis, then vasa efferentia, then kidney. So testis, vasa efferentia, kidney, then Bidder's canal, not seminal vesicle. So first option is incorrect. Then the second one is testis, vasa efferentia, then Bidder's canal. No, before Bidder's canal it will enter the kidney. So this is also incorrect. In the third one we have testis, then vasa efferentia, then kidney, then Bidder's canal, then urogenital duct, and then cloaca. Yes, this is the correct sequence. Okay, so option C will be the correct answer. Question number 48 is from the chapter Reproductive Health, and the question is, in case of a couple where the male is having a very low sperm count, which technique will be suitable for fertilization? Okay, options are gamete intracytoplasmic fallopian transfer, that is written as GIFT in short. Second one is artificial insemination. Third one is intracytoplasmic sperm injection, and the last one is intrauterine transfer. Okay, so the correct answer will be artificial insemination, option number B. So what is artificial insemination? In this method, doctors transfer the sperms directly into the woman's reproductive tract. Okay, so let's write it down here. Doctors transfer the sperms directly into the reproductive tract. That means cervix, fallopian tube, or uterus, depending upon the requirement. Okay. The reason is that the sperm is not powerful enough to move through these parts. Okay. Now let's come to the other options. The first one is GIFT. Okay, gamete intracytoplasmic fallopian transfer. So in this method, the egg is extracted first. Okay. So let's write it down here. Okay. It involves extracting the egg, then fusing them with the sperms, fusing them with sperms, and then inserting them into the fallopian tube. Okay, and then inserting them into the fallopian tube where the egg is fertilized, where the egg is fertilized. Then the third option is intracytoplasmic sperm injection. So what happens in this method? This is an IVF treatment. Okay, this is an IVF treatment that involves injecting a single sperm directly into the egg cell. Okay, that involves injecting a single sperm directly into the cytoplasm of the egg cell. Okay, into the egg cell. Then the last one is intrauterine transfer. So in this method, the sperm is injected directly into the uterus. Okay, intrauterine, that means inside the uterus. So the sperm is transferred into the uterus. So somewhere the sperm is transferred into uterus, somewhere it is transferred into the fallopian tube, somewhere it is transferred directly into the egg cell. So you have to keep in mind the differences. Okay. So according to this question, the correct answer is option B, artificial insemination. Question number 49 is from the chapter Ecosystem, and the question is, which ecosystem has the maximum biomass? Options are grassland ecosystem, pond ecosystem, lake ecosystem, and forest ecosystem. So it is very easy. The correct answer will be forest ecosystem. The reason being the large size and longevity of the trees. Okay. So size and longevity of trees plus the occupied area of the forest ecosystem is very large. Okay, so the area occupied by forest ecosystem is very large. Due to this, the biomass will also be maximum in this case, and as compared to the forest ecosystems, the other options mentioned here have a very low vegetation cover. Okay, very low vegetation cover as compared to the forest ecosystem. Therefore, the correct answer will be option D. Question number 50 is from the chapter Breathing and Exchange of Gases, and the question is, lungs are made up of air-filled sacs, the alveoli. They do not collapse even after forceful expiration because of. So let's discuss one by one. The first one is inspiratory reserve volume, that is IRV. Okay, so this is the additional volume of air that a person can inspire by a forcible inspiration. Okay, so let's write it down here. Additional volume of air that can be inspired by a forcible inspiration. Forcible, we are not talking about the normal inspiration, we are talking about the forcible inspiration. Okay, the additional volume other than the normal volume. Okay, then the second one is tidal volume. So this is the normal volume of air that is inspired or expired during a normal respiration. Okay, so volume of air inspired or expired during a normal respiration. And if there is a forcible respiration, then in addition to tidal volume, inspiratory reserve volume will also be inspired. Okay. Now the third one is expiratory reserve volume. This is just the opposite of inspiratory reserve volume. That means additional volume of air that can be expired. Okay, that can be expired by a forcible expiration. Forcible, this is forcible, okay, forcible expiration. And the last one is residual volume. So as the name suggests, it is the volume of air that remains in the lungs. Volume of air remaining in the lungs, even after a forcible expiration. Okay, even after a forcible expiration. That means no matter how much force you apply during expiration, there will be a certain amount of air that will remain in the lungs, and this amount will prevent the collapse of alveoli. Okay, it prevents the collapse of alveoli. Therefore, the correct answer will be option D, that is residual volume. Question number 51 is from the chapter Ecosystem, and the question is presence of plants arranged into well-defined vertical depending on their height can be seen best in. Options are tropical rainforest, grassland, temperate forest and tropical savanna. So here we are talking about the stratification, isn't it? Stratification. What is stratification? It is the vertical distribution of species. Let's write it down here. Vertical distribution of different species occupying different levels is known as stratification. Okay, and a tropical rainforest is the best example for this. Okay, in this forest the stratification is as follows. Trees occupy the topmost strata. Okay, the topmost strata is occupied by the trees, then shrubs, these are second, and then herbs and grasses, these are the bottommost layer. Okay, bottommost layer, and the trees are the topmost layer. So this is how the vertical distribution is done. Therefore, the correct answer will be option A, tropical rainforest. Question number 52 is from the chapter Excretory Products and Their Elimination. So in this question, the permeability of loop of Henle is discussed. Okay, so suppose this is the loop of Henle. This is the descending loop and this is the ascending loop. Okay, this is the descending loop and this side we have ascending loop. Now this descending loop is permeable to water and the ascending loop is permeable to the electrolytes. Okay, so let's write it down here. Descending loop is permeable to water, but impermeable to the electrolytes, and if we talk about the ascending loop, it will be just opposite. Okay, in ascending loop it will be permeable to the electrolytes, but impermeable to the water. Okay, impermeable to water. Now let's go through the options. The first option says that the descending limb of loop of Henle is impermeable to water. So we have seen that the descending is permeable to water, okay, not impermeable. So this is incorrect. Then the next statement says the ascending limb of loop of Henle is permeable to water. This is again incorrect because the ascending limb is impermeable to water. It is permeable to the electrolytes, right? Then the third statement says that the descending limb of loop of Henle is permeable to electrolytes. So as you can see, the descending is permeable to water and impermeable to electrolytes. So again, this is incorrect. The last statement says that the ascending limb of loop of Henle is impermeable to water. Yes, it is correct because the ascending limb is permeable to electrolytes. It is permeable to electrolytes and impermeable to water. Therefore, option D will be the correct answer. Question number 53 is from the chapter Biodiversity and Conservation, and the question is Alexander von Humboldt described for the first time option A laws of limiting factor, option B species area relationships, option C population growth equation, and option D ecological biodiversity. Okay, so the correct answer is species area relationship. Okay, Alexander von Humboldt was a German naturalist. Okay, he was a German naturalist and geographer, and he described the species area relationship, which states that the species richness within a region increases with increasing explored area. Okay, with increase in explored areas, but only up to a certain limit. Okay, but only up to a certain limit. So the correct answer will be option B, species area relationships. Question number 54 is from the chapter Plant Kingdom, and the question is zygotic meiosis is characteristic of. Options are fucus, funaria, chlamydomonas and mercantia. So the correct answer will be chlamydomonas. Okay, what is zygotic meiosis? It is the meiotic division in the zygote, right? That means the zygote undergoes reduction division after the division of the nucleus, after division of nucleus, that means after karyogamy, okay. So since chlamydomonas is purely haploid plant, therefore in this the zygotic meiosis takes place. So option C will be the correct answer. Question number 55 is from the chapter Molecular Basis of Inheritance, and the question is if there are 999 bases in an RNA that codes for a protein with 333 amino acids and the base at position 901 is deleted such that the length of the RNA becomes 998 bases, how many codons will be altered? Okay, so we know that for every 333 amino acids, 333 amino acids, how many bases are involved? 999 bases are involved in the translation of RNA. Okay, involved in the translation of RNA. Now, it is given that the base at the position 901, that means out of 999, the position 901, here the base is deleted or removed. That means before 901, that means the initial 900 will be remained unaffected. Okay, so initial 900 bases will be normal. The change is done at 901. So the later 99 bases will be modified due to that. Okay, or altered. So how many bases are altered? 99 bases are altered. But the question is asked that how many codons will be altered. Okay, so we know that 3 bases 3 bases make 1 codon, right? 1 codon. So 1 base means 1 by 3 codon. And here 99 bases are altered. So 99 bases means 1 by 3 into 99 codons. So from here we can say that 33, okay, 33 codons are altered. Codons are altered. So this is what is asked here. So option will be, the correct answer will be option number B, 33 codons. Question number 56 is from the chapter Sexual Reproduction in Flowering Plants, and the question is, flowers which have single ovule in the ovary and are packed into inflorescence are usually pollinated by. Options are bee, wind, bat and water. So in such type of flowers, the pollens are lightweight and non-sticky. Okay, lightweight and non-sticky. Therefore, these can easily be pollinated by the wind. Easily pollinated by wind. So which type of pollination will take place? Wind pollination will take place. Okay. And what is the other term for wind pollination? It is known as anemophily. Okay. Anemophily. If we talk about bee, so bee is an insect and insect pollination is known as entomophily. You know this? Entomophily. Then bat. The pollination that is done by bat is known as chiropterophily. Chiropterophily. And that is done by water is known as hydrophily. Okay, very easy. Hydrophily. So here which type of pollination is taking place? Anemophily, that means wind pollination. So option B will be the correct answer. Question number 57 is from the chapter Human Health and Disease, and the question is, transplantation of tissues or organs fails often due to non-acceptance by the patient's body. Which type of immune response is responsible for such rejections? Options are cell-mediated immune response, hormonal immune response, physiological immune response and autoimmune response. Okay, so why does the patient's body not accept the transplanted tissues or organs? Because the immune system of our body treat these transplanted tissues as foreign. Okay, foreign or pathogens. Okay, so these transplanted tissues or organs are treated as foreign pathogens. Okay, therefore the body tends to reject this. Okay, and the major system that is actively involved in this rejection is the T lymphocyte cells. Okay, T lymphocyte cells, which are part of the cell mediated immune response. Okay, cell mediated immune response. Therefore, what will be the correct answer? Option A, cell-mediated immune response will be the correct answer. Question number 58 is from the chapter Plant Kingdom, and the question is, life cycle of ectocarpus and fucus respectively are. Okay, so we know that ectocarpus and fucus are algae. These are examples of algae. Okay, and we know that in most algae, which type of life cycle is there? Haplontic life cycle. Haplontic. That means in these organisms, the haploid gametophyte is predominant stage. Okay, haploid gametophyte is dominant. Now in higher plants, which type of life cycle? Diplontic life cycle. Diplontic. Higher plants means gymnosperms and angiosperms. Gymnosperms and angiosperms. So in these plants you will see diplontic life cycle. That means here the diploid sporophyte stage is dominant. Sporophyte is dominant. and there is a intermediate life cycle also, that is known as haplodiplontic. So in bryophytes, this is important, and pteridophytes, these plants show haplodiplontic life cycle. Haplodiplontic. That means there is an alternation of generation in this case. Alternation of generation between the haploid and diploid stage. Okay. But there are some exceptions in algae. We have seen that most algae have haplontic life cycle, but there are some exceptions. For example, let's write it down here. Exceptions of algae: ectocarpus, then Polysyphonia, and kelps. These are some exceptional algae which show haplodiplontic life cycle. Haplodiplontic. And fucus is also an algae, but it shows diplontic life cycle. Okay, so these are exceptions. So here the question is asked about the ectocarpus and fucus. So the life cycle of ectocarpus will be haplodiplontic and that of fucus will be diplontic. So haplodiplontic and diplontic respectively, that means option B will be the correct answer. Question number 59 is from the chapter Biotechnology, Principles and Processes, and the question is, a gene whose expression helps to identify transformed cells is known as. Options are vector, plasmid, structural gene and selectable marker. So the correct answer is option D, that is selectable marker. Okay, so it is a gene that helps in differentiating the transformants and non-transformant cells. Okay, so let's write it down here. It is a gene in a vector that helps in the selection of transformants. Transformant means the host cells that have successfully taken up the foreign DNA. Okay, and non-transformants Non-transformants means the cells which have not taken up the vector DNA. Okay, so this selectable marker helps in differentiating between them. Now this is resistant to antibiotics. Selectable marker is resistant to antibiotics, and it is used in cloning vectors. Okay, used in cloning vector. So option D will be the correct answer. Question number 60 is from the chapter Sexual Reproduction in Flowering Plants, and the question is, a dioecious flowering plant prevents both. Options are autogamy and getonogamy, getonogamy and xenogamy, cleistogamy and xenogamy, and autogamy and xenogamy. So we know that in case of autogamy, the transfer of pollen takes place from the anther to the stigma of the same flower. Okay, so the transfer of pollen to the same flower or to the stigma of the same flower. In case of getonogamy, the transfer takes place from the anther of one flower to the stigma of different flower, but the plant is same. Okay, so from anther of one flower to the stigma of another flower of the same plant. So in the first case the flower was same, in the second case the plant is same, but in the third case, that is known as xenogamy, both are different. Okay, so different plants. Transfer takes place from the flower of one plant to that of another plant. Okay, different plant. Here in the question, a dioecious flowering plant is mentioned. Dioecious, it means the flowers will be unisexual. Okay, and in case of unisexual flowers, both autogamy and getonogamy is prevented. Okay, so both autogamy and getonogamy is prevented. Only xenogamy is possible. Prevented. So the correct answer will be option A, autogamy and getonogamy. Question number 61 is from the chapter Respiration in Plants, and the question is, which statement is wrong for Krebs cycle? So let's understand the Krebs cycle first. We know that it is also known as tricarboxylic acid cycle or the citric acid cycle. Okay, because the first product that is formed during this cycle is citric acid. Now, before the start of this Krebs cycle, first the pyruvate that was obtained as the final product of glycolysis will get converted into acetyl coenzyme A. Acetyl coenzyme A. So pyruvate is not a part of this TCA cycle, but acetyl coenzyme A is part of the TCA cycle. Okay. Now the first step of the TCA cycle or the Krebs cycle takes place by the condensation of oxaloacetic acid. Oxaloacetic acid, that is a 4-carbon molecule, with the acetyl coenzyme A, that is a 2-carbon molecule. Okay, so we can say that oxaloacetic acid is the first member of this cycle. First member of this cycle. Okay, now 4 carbon plus 2 carbon, that means 6 carbon. So the first product that will be formed will be a 6 carbon molecule, that is citric acid. Okay, therefore it is also known as citric acid cycle. Citric acid, this is a 6 carbon molecule. Then this 6 carbon molecule will get converted into a 5 carbon molecule. So it will lose 1 carbon in the form of carbon dioxide. Also during this step, a NAD plus NAD plus will get reduced to NADH plus H plus. Okay. Now citric acid will get converted into the 5 carbon molecule that is known as alpha ketoglutaric acid. Alpha ketoglutaric acid. This is a 5 carbon molecule. Again, this 5 carbon molecule will lose 1 carbon in the form of carbon dioxide. So it will form a 4 carbon molecule that is known as succinic acid. Okay, succinic acid. So this is a 4 carbon molecule. 4 carbon molecule. Now during this step, two things happen. First, reduction of NAD plus into NADH plus H plus, NADH plus H plus, and second, the conversion of GDP into GTP. Okay, so these two things are important here. Now this succinic acid will not lose any carbon and it will get converted into another 4 carbon molecule that is known as malic acid. Malic acid. So this is also a 4 carbon molecule. During this conversion, the important thing that takes place is the conversion of FAD plus into the FADH plus FADH2. Okay, FADH2. So this is important. There is only one step where FAD plus is converted into FADH2, that is during the conversion of succinic acid into malic acid. Okay, then finally malic acid is converted into oxaloacetic acid, and here also the reduction of NAD plus takes place to NADH plus H plus. Okay. So now let's see the statements. In the first statement, it is mentioned that there is one point in the cycle where FAD plus is reduced to FADH2. So yes, we have seen there is only one point during the conversion of succinic acid into malic acid where FAD plus is converted into FADH2. Okay, only one point. Therefore, this is a correct statement. Then the second statement says during conversion of succinyl coenzyme A to succinic acid, a molecule of GTP is synthesized. So this is also correct. See, during the conversion of succinic acid, see between alpha ketoglutaric acid and succinic acid, there must be the formation of succinyl coenzyme A that we have not dealt in detail. But during this conversion, or you can say that during the formation of succinic acid, a molecule of GTP is synthesized. Okay, there is a conversion of GDP into GTP. So this is also correct. We have seen here. So this statement is also correct statement. Now let's come to the third statement. The cycle starts with condensation of acetyl coenzyme A with pyruvic acid to yield citric acid. This is absolutely incorrect because citric acid was formed by the condensation of oxaloacetic acid and acetyl coenzyme A. Okay, pyruvate was not part of the cycle. Okay, so this is incorrect statement. And the last statement says there are 3 points in the cycle where NAD plus is reduced to NADH plus H plus. Okay, so this is also correct. See, this is the first point from during the conversion of citric acid into alpha ketoglutric acid. Then the second during the conversion of alpha ketoglutric acid into succinic acid, and third during the conversion of malic acid into oxaloacetic acid. Okay, so 3 points in the cycle where this thing happens. So this is also correct statement. So we had to find the wrong statement. So the only wrong statement is the third one. Option number C will be the correct answer. Question number 62 is from the chapter Photosynthesis in Higher Plants, and the question is phosphoenol pyruvate, that is PEP, is the primary carbon dioxide acceptor in. Options are C4 plants, C2 plants, C3 and C4 plants, and C3 plants. Okay, so the correct answer is C4 plants. In C4 plants, the primary carbon dioxide acceptor is phosphoenol pyruvate, that is PEP, and it is a 3 carbon molecule. Okay, so it accepts the carbon dioxide, that is a 1 carbon molecule, and it forms 3 plus 1, that is 4 carbon molecule, that is oxaloacetic acid. Okay, this is a 4 carbon molecule, and this reaction is catalyzed by the enzyme. Which enzyme? The pepcase enzyme. Pepcase enzyme catalyzes this reaction. Okay, now if we talk about the C3 plants, here the primary carbon dioxide acceptor is RUBP. RUBP, that is ribulose bisphosphate, which is a 5 carbon molecule. So it accepts the carbon dioxide, which is a 1 carbon molecule. So will it form a 6 carbon molecule? No, it will form 2 molecules containing 3 carbons each. Okay, 2 molecules containing 3 carbon each, and these 3 carbon molecules are 3-PGA, phosphoglyceric acid. Okay, that is a 3C3 acid, and this reaction is catalyzed by which enzyme? In this case the enzyme is Rubisco enzyme. Okay, the most abundant enzyme. Okay, the full form is RUBP, carboxylase oxygenase. Okay, so here the question was asked about the PEP. So this is the primary carbon dioxide acceptor in C4 plants. Therefore option A will be the correct answer. Question number 63 is from the chapter Molecular Basis of Inheritance, and the question is during DNA replication Okazaki fragments are used to elongate. Okay, so let's see the DNA replication fork. Suppose this is the DNA strand. It is separated here for replication. Okay, this is the 5' end, this is the 3' end. Here we have 3' end and this is the 5' end. Okay, now we know that DNA replication always takes place in the direction 5' to 3'. Okay, so it will start with 5' towards the 3'. Okay, so it will start from here. This is the 5', so the replication will start in 5' to 3' direction. Okay, and this strand that will be formed as the result of the replication will be a continuous strand. That is also known as leading strand. Okay, leading strand, because this replication takes place first. Okay, then the second side of this replication fork, here the replication will be discontinuous in the opposite direction. Okay, in this direction, like this. Okay, so this is a discontinuous strand, continuous strand, that is also known as lagging strand. Okay, lagging strand, and these fragments are known as Okazaki fragments. Okay, these are known as Okazaki fragments. So we can see that these fragments are used to elongate which strand? Lagging strand. And in which direction? Away from the replication fork. Okay, in this direction. So the first option is the lagging strand towards the replication fork. No, replication fork is this side, so it is not towards this side, it is away from it. Then the second option is the leading strand, it is not leading strand. Okay, third one is lagging strand away from the replication fork. Yes, so option C will be the correct answer. Question number 64 is from the chapter Molecular Basis of Inheritance again, and the question is, which of the following RNAs should be most abundant in animal cell? Options are tRNA, mRNA, miRNA and rRNA. So the correct answer will be rRNA. Okay, it forms 80% of the total RNA content. Okay, of the total RNA content of a cell. This is occupied by which RNA? By the rRNA. And this rRNA is a single-stranded and shows the presence of intra-chain hydrogen bond. The presence of intra-chain hydrogen bond. Now this rRNA is part of ribosome, right? It is part of ribosome, and we know that ribosome is the site for protein synthesis. For protein synthesis. So rRNA is part of protein synthesis. Therefore, it is the most abundant RNA in the animal cell. Therefore, option D will be the correct answer. Question number 65 is from the chapter Chemical Control and Coordination, and the question is, gonadotrophin releasing hormone, a hypothalamic hormone needed in reproduction, acts on. Okay, so gonadotrophin releasing hormone, it is related to gonads. Okay, it helps in the development of the gonads, development of gonads, and it acts on which part of the pituitary gland? The anterior part of the pituitary gland. Okay, acts on anterior pituitary. Then it stimulates the secretion of two hormones, mainly LH, that is luteinizing hormone, and FSH, that is follicle stimulating hormone. These two hormones regulate the reproductive activity, reproductive activity in male and female. Okay, male and female. So let's see the option. The first option is this gonadotrophin releasing hormone acts on anterior pituitary gland and stimulates the secretion of luteinizing hormone and follicle stimulating hormone. We have discussed, this is the correct answer. Okay. Second and third option are incorrect because it says it acts on posterior pituitary, which is not true. Okay, it acts on anterior pituitary. Then the last one is anterior pituitary gland and stimulates the secretion of luteinizing hormone and oxytocin. No, oxytocin is not involved here. So only correct option will be option number A. Question number 66 is from the chapter Biotechnology, Principles and Processes. And the question is, what is the criterion for DNA fragment movements on agarose gel during gel electrophoresis? So in one of the previous questions, we have already discussed about the agarose gel electrophoresis technique. Okay, we know that the DNA fragments will be positively charged or negatively charged. These are negatively charged. Okay, negatively charged. DNA is a negatively charged molecule. Now these are separated on the basis of size. These are separated on the basis of size. And here the smaller sized fragment will move farther. It moves farther. So let's see the options. The first one says that the smaller the fragment size, farther it moves. Yes, it is a correct statement. Okay. Second one says positively charged fragments move to farther sides. So we have seen that DNA is a negatively charged molecule. Okay. So the fragments are also negatively charged, not positively charged. So this is incorrect. Third one says negatively charged fragments do not move. It is also incorrect because negatively charged fragments move on the basis of size. Okay. The last one says the larger the fragment size, the farther it moves. No, smaller the fragment size, the farther it will move. So the only correct statement is option number A. Okay. Question number 67 is from the chapter Chemical Control and Coordination, and the question is hypersecretion of growth hormone in adults does not cause further increase in height because option A, epiphyseal plates close after adolescence, option B, bones lose their sensitivity to growth hormone in adults, option C, muscle fibers do not grow in size after birth, and option D, growth hormone becomes inactive in adults. Okay. So the correct answer is option A, that is epiphyseal plates close after adolescence. Okay. We know that growth hormone is produced by which part of the brain? By the anterior pituitary gland. Anterior pituitary. Now, as the amount of growth hormone increases, the height will also increase, but only up to a certain limit. If after that limit there is hypersecretion of growth hormone, okay, that means more amount than required, then the height will not increase. And the reason is that epiphyseal plates, epiphyseal plate. So this plate closes after adolescence. So what is epiphyseal plate? It is a hyaline cartilage plate. It is a hyaline cartilage plate that is present at each end of the long bone. Okay, plate at each end of a long bone where new bone growth takes place. Okay, where bone growth takes place. So suppose this is the bone where the growth is taking place. Okay, this bone is growing in height. So at both the ends there is epiphyseal plates, but initially they are open. Okay, then the height will go on increasing, but once it closes during adolescence, after adolescence, then no further increase will take place. Okay, so therefore option A will be the correct answer. Question number 68 is related to the chapter cell cycle and cell division, and the question is DNA replication in bacteria occurs. Option A, within nucleolus, option B, prior to fission, option C, just before transcription, and option D, during S phase. So the correct answer will be option B, that is prior to fission. Okay, so bacteria is a prokaryote, right? It is a prokaryote, and in prokaryotes the DNA replication takes place prior to the fission, prior to fission. Whereas if we talk about the eukaryotes, in case of eukaryotes the DNA replication takes place during the synthesis phase or during S phase, right? So for this question the correct answer will be option B. Question number 69 is from the chapter Principles of Inheritance and Variation, and the question is which one from those given below is the period for Mendel's hybridization experiments? Okay, so this is a very easy and factual question. Okay, Mendel performed his hybridization experiments on garden pea. We know that garden pea, that is known as Pisum sativum. Pisum sativum. And the time period of his experiments was 1856 to 1863. Okay, therefore the correct answer will be option D. Question number 70 is from the chapter Biological Classification, and the question is viroids differ from viruses in having. Okay, so let's have some information regarding viroids. So we know that viroids were discovered in the year 1971. Okay, discovered in the year 1971 by the scientist named T.O. Diener. T.O. Diener. Okay. Now, it was mainly smaller than the viruses. Okay, in size they were smaller than viruses. And they were found to be free RNA. Okay, free RNA without protein coat. Okay, free RNA which lacked protein coat. So this was a difference between the viroids and viruses. Okay, because viruses had protein code but viroids did not have protein code. Okay, they were free RNA. Okay, and another important characteristic was that they had very low molecular weight. Okay, so the RNA of viroid was of low molecular weight. So let's see the options now. The first option is viroids differ from viruses in having DNA molecules without protein code. So we have seen that viroids are free RNA, not DNA. Okay, so this is incorrect. Then the second one is RNA molecules with protein code. So this is also incorrect because they did not have protein code. Then the third one is RNA molecules without protein code. Yes, this is the correct one. Last one says again DNA, so incorrect. So the only correct one is option number C. Question number 71 is from the chapter Human Health and Disease, and the question is malt constitutes about -% of the lymphoid tissue in human body. So what is the full form of malt? It is mucosa associated lymphoid tissue. So as the name suggests, it is a network of lymphoid cell that is associated with the mucosal glands or submucosal layers. Okay, so let's write it down here. It is a lymphoid cell network found in submucosal layers of various tracts of our body, such as GI tract, GI, that is gastrointestinal tract, vaginal tract, respiratory tract, urinary tract, etc. Okay, systems, etc. Now, the function of malt is that it generates immunological responses. Okay, so it generates immunological responses to antigens found in these mucosal surfaces. Okay, found on all the mucosal surfaces because it is associated with mucosa only. Okay, so mucosal surfaces, and in the human body it makes up 50% of the lymphoid tissue. Okay, it makes up 50%, about 50%, that means about half of the lymphoid tissue, in humans or human body. So which will be the correct answer? Option D, 50% will be the correct answer. Question number 72 is from the chapter Microbes in Human Welfare, and the question is which of the following is correctly matched for the product obtained by them. Okay, so let's see. In the first one, methanobacterium is matched with lactic acid, which is an incorrect match. Okay, so if we talk about methanobacterium, it is an anaerobic bacteria. Okay, and it helps in the fermentation of organic matter. So it helps in anaerobic fermentation of organic matter, and thus it is responsible for the production of biogas. Therefore it is a incorrect match. Okay, if we talk about the lactic acid, it is produced by which bacteria? Lactobacillus species. Okay, this is very common, lactobacillus. Now let's come to the second one. In the second one, penicillium notatum is matched with acetic acid, which is again incorrect. If we talk about the penicillium notatum, as the name suggests, it is responsible for the production of penicillin, which is an antibiotic, right? Antibiotic. So this is the source of antibiotic whereas acetic acid is produced by the Acetobacter aceti. Acetobacter aceti. Okay, therefore this is also incorrect. Now let's come to the third one. Saccharomyces cerevisiae is matched with ethanol. Yes, this is a correct match. Okay, Saccharomyces cerevisiae, it is an yeast. Okay, yeast, and that is a unicellular fungus. Unicellular fungus. Okay, and it produces the ethanol by the fermentation of sugar. Fermentation of sugar. So after fermentation, the product obtained is ethanol. Product is ethanol. Therefore, this is a correct match. Now let's come to the last one. Here Acetobacter aceti is matched with antibiotics. That is incorrect. We have seen that Acetobacter aceti is responsible for the production of acetic acid. Okay, and antibiotics are produced by, specifically penicillin is produced by the Penicillium notatum. Okay, so the correct match is option number C. Question number 73 is again from the chapter Biological Classification, and the question is, which among the following are the smallest living cells known without a definite cell wall, pathogenic to plants as well as animals, and can survive without oxygen? Okay, options are Pseudomonas, Mycoplasma, Nostoc, and Bacillus. So the correct answer is option B, that is Mycoplasma. These are the smallest living cells known. Okay, and all the information regarding mycoplasma is already mentioned in the question. These are smallest living cells known. They completely lack a cell wall. Okay, then these are pathogenic to plants as well as animals, and they can survive even without oxygen. Okay, so option B will be the correct answer. Okay. Question number 74 is from the chapter Biological Classification, or you can relate it to the animal kingdom. Okay, and the question is, which of the following represents the order of horse? Options are Perissodactyla, Caballus, Ferus, and Equidae. So let's see, what is the scientific name of horse? Its name is Equus caballus. Okay, Equus caballus. So Equus is the genus. Okay, and Caballus is the species. Okay, now if we talk about the phylum, it is clear that the horse belong to Chordata phylum. Class is Mammalia. Order is Perissodactyla. Perissodactyla. Family is Equidae. So Equidae is also mentioned in the options, which is the name of the family. Okay, so genus and species we have already discussed. It is Equus and Caballus respectively, but the question is asked about the order. So order is Perissodactyla, this one. Okay, therefore correct answer will be option number A. Question number 75 is from the chapter Structural Organization in Animals, and the question is, frog's heart when taken out of the body continues to beat for some time. Select the best option from the following statements. Okay, now first statement is frog is a poikilotherm. Second one is frog does not have any coronary circulation. Then the third one is heart is myogenic in nature, and the last one is heart is autoexcitable. See, the statements in individually are all correct. Okay, but the first two statements are not the correct reason for this. Okay, when the heart is taken out of the body, it continues to beat. So these two statements are not the correct reason for that. The correct reason will be the third and fourth statement. Okay, heart is myogenic in nature. So heart is myogenic means what? These are originated in muscle tissue. Okay, so heart of frog are myogenic in nature, which means that originates it originates in the muscle tissue muscle tissue and not from the nerve impulse. Okay, and second important thing is that it is auto excitable. It is auto excitable. That means it does not need need external stimuli. External stimuli. So when frog's heart is taken out of the body, it will continue to beat for some time. That is till the time ATP is available. Okay. So it will beat for some time till the ATP is available. Till the ATP is available. Because it is not dependent on the nervous system and it does not require external stimuli. Okay. So the correct answer will be C and D. That means option C. Okay. Option C will be the correct answer. Question number 76 is from the chapter Strategies for the Enhancement in Food Production. And the question is homozygous pure lines in cattle can be obtained by. Okay, options are mating of unrelated individuals of same breed. Option B, mating of individuals of different breed. Option C, mating of individuals of different species. And option D, mating of related individuals of same breed. So the correct answer will be option D, that is mating of related individuals of same breed. This process is known as inbreeding. Okay, inbreeding. That is mating of related individuals of the same breed. Okay. Now inbreeding increases the homozygosity. Inbreeding increases homozygosity and it is responsible for pure lines production in cattle. Okay. Responsible for pure lines production in cattle. Therefore, the correct answer will be option number D. Question number 77 is from the chapter Anatomy of Flowering Plants and the question is identify the wrong statements in context of heartwood. So what is a heartwood? It is the region of the stem. Let's write it down here. It is the region of the stem that contains dead elements. Okay, dead elements with highly lignified walls. Highly lignified, that means they contain lignin. Okay, lignified walls. Okay, these are known as heartwood. Now let's see the option. The first option, it is highly durable. Yes, it is highly durable. Okay, then the second statement says it conducts water and minerals efficiently. No, this is incorrect because we have seen that they contain dead elements. Okay, so due to the presence of dead elements, they are not able to conduct water and minerals efficiently. So what is their function? The main function is to provide the mechanical support. Okay, mechanical support. So although they are not able to conduct, but they can provide the mechanical support to the plants. Third statement is it comprises dead elements with highly lignified walls. This is correct. Okay. Then the last statement says organic compounds are deposited in it. Yes. So organic compound, that means we are talking about the lignin. Lignin are the organic polymer. Okay. Lignin are organic compounds. These are deposited. Therefore, these are known to be lignified. Okay. So the only incorrect or the wrong statement is statement B. So option B will be the correct answer. Question number 78 is related to cell cycle and cell division. And the question is anaphase promoting complex, that is APC, is a protein degradation machinery necessary for proper mitosis of animal cells. If APC is defective in human cell, which of the following is expected to occur? Option A, chromosomes will be fragmented. Option B, chromosomes will not segregate. Option C, recombination of chromosome arms will occur. And option D, chromosomes will not condense. Okay. So as the name suggests, anaphase promoting complex, that means this APC helps in anaphase. Okay, helps during the anaphase stage of the mitosis. Okay, and what happens during anaphase? The chromatids separate. Okay, so the separation of chromatids takes place during the anaphase. And this anaphase promoting complex helps in the separation of chromatids during the anaphase. Okay, we know that chromatids will start moving towards the opposite pole. So if this APC is defective in human cell, that means it will affect this anaphase. Okay, so it will affect the separation of chromatids. Okay, so the chromatids will not segregate. Okay, chromatids will not separate, or we can say the chromosomes will not segregate. Therefore, option B will be the correct answer. Question number 79 is from the chapter Cell, the Unit of Life, and the question is, which of the following cell organelles is responsible for extracting energy from carbohydrates to form ATP? Options are ribosomes, chloroplast, mitochondrion and lysosomes. So this is a very easy question. We know that ATP, that is also known as energy currency of the cell, energy currency of the cell is formed in the mitochondria. Okay, so mitochondria helps in extracting energy from carbohydrates to form the ATP. Okay, it is formed by the mitochondria. Therefore, mitochondria is known as the powerhouse of the cell. Powerhouse of the cell. Right? Ribosome is responsible for protein synthesis. We all know it is responsible for protein synthesis. Chloroplast helps during the photosynthesis. And lysosome has digestive enzymes. Okay, so it helps in digestion. So according to the question, the correct answer will be option C, mitochondrion. Now question number 80 is from the chapter Biological Classification, and the question is, mycorrhizae are the example of. Options are amensalism, antibiosis, mutualism and fungistasis. Okay, so the correct answer is option C, that is mutualism. It is a very common example of mutualism, right? Here there is a symbiotic association between fungi and roots of higher plants. Okay, roots of higher plants. This is known as mycorrhizae. Therefore, option C will be the correct answer. Question number 81 is from the chapter Locomotion and Movement, and the question is, out of X pairs of ribs in humans, only Y pairs are true ribs. Select the option that correctly represents the values of X and Y and provides their explanation. Okay, so we know that out of the 12 pair of ribs in humans, the first 7 pairs are known as true ribs. First 7 pair are known as true ribs, and the reason is that dorsally they are connected with the vertebral column, whereas ventrally they are connected directly to the sternum. Okay, to the sternum. Okay, so they have articulation on both the sides. After 7th, that means 8th pair, 9th pair and the 10th pair, these are known as false ribs. Okay, these are known as false ribs or vertebrochondral ribs. The reason is that they are not directly connected to the sternum. Okay, not directly connected to the sternum on the ventral side. Okay, instead they are connected to the 7th rib. They are connected to the 7th rib. So this is how they indirectly connect with the sternum. Got it? And after that, 11th and 12th pair, 11th and 12th pair are known as floating ribs, because these are neither directly nor indirectly connected to the sternum. Okay, so 3 types of pairs are there. First is the true ribs, second type is false ribs, and third type is floating ribs. Okay, so here out of the X pairs, that means X is the total number, that means 12 pairs. Okay, only Y pairs are true ribs. So how many true ribs are there? How many pairs? 7 pairs. Okay, first 7 pairs are true ribs. So let's see. The first option is X equals to 12, Y equals to 5. So this is incorrect because Y equals to 7. Okay, then the second one is X is 24. So total is only 12, not 24. So this is incorrect. Then the third one is X equals to 24. Again incorrect. Then the last one is X equals to 12. Yes, total number of total pairs of ribs are 12, and y equals to 7. So 7 are true ribs. That is also correct. The reason is given. True ribs are attached dorsally to the vertebral column and ventrally to the sternum. Okay, so this is absolutely correct. So option D will be the correct answer. Question number 82 is from the chapter Animal Kingdom, and the question is, in case of poriferans, the spongiocele is lined with flagellated cells called. Okay, so we know that porifera are sessile animals. Okay, these are aquatic animals, right? They have minute pores on their bodies. These pores are known as ostia. And then there is a large pore here that is known as osculum. So the water enters through ostia and exits through the osculum. Okay, but before exiting through the osculum, it moves into the cavity, inner cavity. Okay, so that is known as spongiocele. Body cavity, inner body cavity is known as spongiocele. Spongiocele, and this body cavity, spongiocele, is lined with flagellated cells called choanocytes. Choanocytes. Therefore, the correct answer will be option B, choanocyte cells. Okay. Question number 83 is from the chapter Environmental Issues, and the question is, which one of the following statements is not valid for aerosols? Option A, they alter the rainfall and monsoon patterns. This is a correct statement. Option B, they cause increased agricultural productivity. This is incorrect. Okay, aerosols are harmful to our environment. These are harmful to the environment. So these are not beneficial, but here this statement says that they are beneficial agriculturally. So this is incorrect. Then the third one is they have negative impact on the agricultural land. Yes, this is correct. Then the last one is they are harmful to human health. Okay, so they have only negative and harmful effects, not a positive effect. Therefore, option B will be the correct answer. It is not valid. Question number 84 is from the chapter Digestion and Absorption, and the question is, a baby boy aged 2 years is admitted to play school and passes through a dental checkup. The dentist observed that the boy had 20 teeth. Which teeth were absent? Options are canines, premolars, molars and incisors. So if we talk about the dental formula, so in adult the dental formula is 2123. This is for upper jaw, and upon 2123. This is for lower jaw. Okay. So we are talking about the each half of the upper and lower jaw. If we talk about the whole complete jaw, then it will be 2 times. Okay. But in children of about 2 years, the dental formula will be 2102 2102. Okay, so the number of incisors and canine, the first one is incisor, second one represents canines, third one represents premolars, and the fourth one represents molars. So as we can see that the number of incisors and canines are same in adult and children. Okay, but there is no premolar in children. No premolar. So premolars are absent. Even they have the molars, that is less in number as compared to the adult, but it is present. Okay, so premolars will be absent. That means option B will be the correct answer. Question number 85 is from the chapter Plant Kingdom, and the question is, select the mismatch. Okay, so in the first one, cycas is matched with dioecious. Now we know that cycas is a gymnosperm plant. Here the males and females cones are present on different trees. Okay, male and female cones or strobili are present on different trees or different plants. So cycas is dioecious in nature. So this is a correct match. Okay. Then the second one, Salvinia is matched with heterosporus. This is also a correct match because Salvinia produces spores of different types. Okay. Spores of different types are produced by the Salvinia. Therefore heterosporous. Third one, Equisetum homosporous. Yes, Equisetum, in Equisetum the spores of same types are produced. Okay, same types are produced, so it is homosporous in nature. Then the last one, Pinus is matched with dioecious. So Pinus is also a gymnosperm and it has both male and female, both male and female cones on the same plant. Okay, on the same plant. So it is monoecious in nature. Monoecious, not dioecious. Therefore, this is a mismatch. Okay, this is incorrect match. So option D will be the correct answer. Question number 86 is from the chapter Sexual Reproduction in Flowering Plants, and the question is the morphological nature of edible part of coconut is. Options are cotyledon, endosperm, pericarp and perisperm. So this is very easy. We have already discussed about coconut many times. Okay, we know that the edible part of the coconut is nothing but the endosperm. Okay, so option B is the correct answer. Question number 87 is again from the chapter Sexual Reproduction in Flowering Plants, and the question is double fertilization is exhibited by. Options are algae, fungi, angiosperms and gymnosperms. So we know that double fertilization is a unique characteristic that is found only in the angiosperms. Okay, so option C is the correct answer. What happens in double fertilization? Here two male gametes take part in the fertilization. Two male gametes are involved. So one male gamete fuses with the or fertilizes with the egg to form diploid zygote. Okay, diploid zygote. The other male gamete fuses with two polar nuclei to form triploid endosperm. Triploid endosperm. Okay, so two fertilizations are taking place, therefore it is known as double fertilization. Okay, option C will be the correct answer. Question number 88 is related to molecular basis of inheritance, and the question is spliceosomes are not found in the cells of. Options are fungi, animals, bacteria and plants. So what are spliceosomes? Let's write it down here. Spliceosomes. See, these are large molecules complex which are found in the nucleus of eukaryotic cells. Okay, nucleus of eukaryotic cells, and they play important part in the splicing of introns. Okay, so it is assembled from snRNAs and protein complexes that plays an important role in the splicing of introns. Okay, in splicing of introns. So as we have seen that these are present in the nucleus of eukaryotic cells. So here we can see that fungi, animals and plants are eukaryotes, whereas bacteria is a prokaryote. It is not a eukaryote, therefore these are not present in the bacteria. Okay, so option C will be the correct answer. Now question number 89 is again from the chapter Molecular Basis of Inheritance, and the question is the association of histone H1 with a nucleosome indicates. Okay, option A, DNA replication is occurring. Option B, the DNA is condensed into a chromatin fiber. Option C, the DNA double helix is exposed, and option D, transcription is occurring. Okay, so we know that the negatively charged DNA, DNA wraps around the histone octamer. Okay, histone octamer, which is positively charged. Okay, this histone octamer is positively charged and it forms the nucleosome. Okay, it forms nucleosome. Here we can put a plus sign. Okay, so negatively charged DNA wrapped around the histone octamer forms a nucleosome. Now the H1 protein binds to the linker DNA, binds to the linker DNA region between the nucleosomes, Suppose this is one nucleosome in which the negatively charged DNA is wrapped around the histone octamer. This is another nucleosome. Okay, so here these are linked together. Okay, H1 protein is responsible for this binding. Okay, so this association of H1 with the nucleosome packs the DNA into the chromatin fiber. Okay, like this chromatin fiber is made. Okay, so the correct answer will be option B, that it indicates that the DNA is condensed into a chromatin fiber. Option B will be the correct answer. And now the last question is from the chapter Biodiversity and Conservation, and the question is, the region of biosphere reserve which is legally protected and where no human activity is allowed is known as. Options are buffer zone, transition zone, restoration zone and core zone. Okay, so we know that in a biosphere reserve there are three main zones. The innermost zone is known as the core zone. Okay, so this is the core zone. Core zone is the innermost zone, and in this zone no human activity is allowed. Okay, no human activity is allowed. Outside the core zone we have buffer zone. So this shaded portion is the buffer zone. Okay, buffer zone surrounds the core zone, and here only limited activities are allowed. Only limited activities allowed. And the outermost zone is known as the transition zone. So this is the transition zone. Transition zone. Now this is known as zone of cooperation. So here various cooperative activities are done, such as settlement, croplands, managed forests, etc. Okay, so here the question is asked about that zone where no human activity is allowed. Okay, so that is none other than the innermost zone, that is the core zone. Therefore the correct answer will be option number D. Okay, so here we complete all the biology questions from NEET 2017 paper. We'll be back with another session soon. Till then, keep learning, keep scoring high. For a better understanding, I would suggest you to practice same questions from our website www.examsnet.com, and for more information, download our app from the link given in the description box. Thank you so much.